Welcome to GIST 101, Understanding the Basics in Biology Behind GIST. Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild. I'm the Allergy Program Director, and I am your moderator for today. I'm going to hand off a brief announcement by Erin Christoph, our Communications Director.
Hi everyone, thank you for joining us today. I just wanted to pop on and let everybody know, as a reminder, today is Rare 13th Day. In case anyone doesn't know, we launched it last year on August 13th to celebrate the 13 people diagnosed each day in the United States with GIST. We've done a lot of great things in the past to commemorate it, and if you don't know, this is what GIST Awareness Day was born from. So today we're just encouraging people to get awareness out there. You can go to therare13.com and learn about GIST, ways to celebrate, and read about some of the real faces of GIST. If you're into that sort of thing, you can hashtag therare13. We love to see videos and pictures of people just really enjoying, playing with the number 13 and celebrating us and awareness for GIST. So have a great webcast, everybody.
Thank you, Erin. So now I'd like to introduce David Josephy. He's a good friend of the Life Raft group. He's a professor at the University of Guelph in Ontario, Canada, where he teaches biochemistry and molecular biology. His sister-in-law has been living with GIST for more than a decade, also a good friend of ours. And David has published more than 100 peer-reviewed scientific papers in biochemistry and cancer research, and he is the co-author of the textbook Molecular Toxicology. In 2010 to 2011, he spent a sabbatical leave in the GIST research lab of Dr. Jonathan Fletcher at Harvard Medical School. David is the president of Life Raft Group Canada, and he is also on the allergy science team. I'd also like to announce that this webcast is dedicated to Mike Burns and Dave Stafford. I would like to now hand off the presentation to David Josephy. David, you may begin.
Thank you, Sarah, and thanks for arranging it, and welcome everyone. So I'll begin by pointing out that I'm not a doctor, I'm not a medical doctor, I'm a scientist, and so I will touch on some medical topics, but for real medical answers, you have to talk to your physician, as Sarah has already pointed out. So in this seminar, I wanted to tell you a little bit about what GIST is as a disease, briefly about what causes GIST. I will talk about how GIST differs from other more common gastrointestinal cancers. Then we will get into the biology of GIST and talk about where, that is, the type of cells in which GISTs arise. I'll mention the difference between localized and metastatic disease. We'll say a little bit about how GIST is treated. We'll talk about some of the mutations underlying GIST, terms you may have heard like KIT and PDGFRA, and we'll say a little bit about how the drugs work. So what is GIST? As I think most people on the call will know, it's a rare cancer. There are about 5,000 cases per year newly diagnosed in the USA. The incidence is about the same everywhere that has been studied in the world. So it seems to be quite a constant incidence around the world, but a rare disease. So GIST is a sarcoma. This is a rare class of cancers. Only about 1% of all cancers are sarcomas. We'll talk about what that means a little later. And in fact, of all the gastrointestinal cancers, only about 1% of them are GIST. So the disease is rare, and as I will explain again later in the talk, it was until about the year 2000, it was a poorly understood form of cancer, and in fact was very often misdiagnosed. Since the year 2000, a lot of progress has been made both in understanding the biology of GIST and also in the treatment of GIST. So first of all, what causes GIST? The question that everyone affected by the disease immediately asks, understandably. What did I do that might have caused the disease? We know that GIST can in fact be inherited. That is, there are families in which many members of the same family have had GIST. This is exceedingly rare. It's a matter of a handful of cases, even in the entire USA. So it's very rare for GIST to be an inherited disease. Almost all GISTs are in fact what we call sporadic cases. That is, they do not run in the family, they're not inherited genetically. There are no known causes of GIST. Nothing in the environment, nothing occupational, nothing in diet or lifestyle, cigarette smoking or alcohol or whatever. There's simply no known causes of GIST. As far as we can tell, it strikes completely at random. And in fact, if there were any significant environmental or lifestyle causes of GIST, I think we would surely know that by now. People even attending GIST meetings like the Life Fest would have realized that they had certain exposures in common, but they don't. So as far as we know, there's no environmental cause to GIST. Now, I said earlier that GIST is a sarcoma. This distinguishes GIST from almost all cancers. Almost all cancers, 99%, are what we call carcinomas. Carcinomas are diseases that arise in what we call epithelial tissues. These are the tissues that line either the external or internal surfaces, the linings that we find inside or within the gastrointestinal tract. So, for example, breast cancer and skin cancer really arise in surface lining tissues on the surface of the body. Colon cancer, bladder cancer, these are lung cancer, these all arise in lining type tissues. They're called carcinomas. But GIST is not of that nature. It's what we call a sarcoma. Sarcomas are cancers that arise from, in general, connective tissues or internal tissues within the body, like blood vessels, cartilage, or bone. In particular, if we look at the stomach, which is where many GISTs arise, a carcinoma begins on the inside, that is, the inside lining of the gastrointestinal tract. GISTs, on the other hand, begin outside the stomach, in the muscular wall that surrounds the stomach. For a long time, it was not at all clear what cell type is the origin of GIST, where the GISTs actually begin. But now we know from genetic and biological studies. So GIST begin in the so-called ICCs, the interstitial cells of Cajal. This is a kind of cell that was first identified by a Spanish biologist named Ramon y Cajal. These are a particular, unique kind of cell that lines the gastrointestinal tract. So GIST begin either in these cells or in very closely related cells, the ICCs. So what are these cells? Well, when you eat food, when you digest, the food is pushed gradually along the gastrointestinal tract, from the esophagus right down to the anus. And that pushing is the result of coordinated waves of muscle contraction all the way along the GI tract. So there are smooth muscle cells that surround each portion of the GI tract, the stomach, the colon, all the way along. To carry out this peristalsis or pushing motion, those contractions of those muscles have to be coordinated. They have to be synchronized. The ICCs are the pacemakers. They're the cells that send out a signal, an electrical signal, that coordinates these waves of muscle contraction. So they're what we call pacemaker cells. And they occur all the way along the GI tract, and that's why GISTs can arise at any point along the GI tract. So I want to stress that all GIST tumors have something very much in common. They arise in the same kind of cell. And that's true essentially regardless of where they arise along the GI tract. There are some biological differences, but they're minor. The fundamental similarity is that all of these tumors arise in the ICCs, whether they occur in the esophagus or most commonly in the stomach or less commonly in the small intestine or rectum. So it's very important, I want to make this an important take-home message from the talk, that all GISTs are GISTs. It's the kind of cell in which they arise, the ICC cell, that defines a GIST, not the anatomical location. So if a GIST begins in the wall of the stomach, it's still a GIST. It's not a stomach cancer as people usually refer to it. When people say stomach cancer, they're usually referring to the common carcinoma that arises in the stomach, the adenocarcinoma. If it just starts in the colon, it's a GIST. It's not colon cancer. When people say colon cancer, they're usually referring to a colorectal carcinoma, a far more common disease. So a GIST is a GIST regardless of the organ where it arises. A GIST is a cancer, and like all cancers, GIST is a dangerous disease. Why? Well, there are many reasons. First of all, the local growth of the tumor growing in the original site, such as the stomach or the colon, can cause problems. It can squeeze on other organs. It can disrupt the function of the GI tract. It can bleed. These are all serious issues. Secondly, like all cancers, GIST can metastasize. That's a very important concept that we'll return to a few times during the talk. Metastasis means that individual cells are dislodged or moved from the original tumor, what we call the primary, and they spread through the bloodstream, through the circulation. They lodge in distant sites in the body, such as the liver, and they begin to grow in those sites. That's a metastasis or a MET. Finally, the cancer itself can lead to complications such as anemia or immune system dysfunction. And unfortunately, many cancer treatments can also cause life-threatening complications such as susceptibility to infection. So how is GIST diagnosed? What's the common symptom that people observe that leads them to go to the doctor, leading to a GIST diagnosis? The answer is there is no typical symptom. In fact, GISTs can grow locally to surprisingly large before they produce any symptoms at all. Common symptoms that are reported include, for example, feelings of discomfort in the abdomen. The problem is that many of these symptoms are also can be caused by many other much more common conditions. It's unfortunate because it means that GIST diagnosis, regrettably, is often delayed because doctors don't recognize it, they're confused, they think that it's some other more common condition. So here's a direct quote that we heard from a patient in Canada. After a whole year during which he kept going back to his family doctor saying he didn't feel well, and the doctor kept on telling him he just had a bad case of heartburn. Finally, he got to another doctor. The second doctor did more extensive tests and recognized that he had GIST. So this is unfortunately common, that GISTs are often delayed in their diagnosis. Now, returning again to this very important issue of localized versus metastatic disease. Sometimes when a GIST is recognized by a doctor when it's diagnosed, sometimes that happens while the disease is still localized. Sometimes it does not happen until the disease has already metastasized. In any case, once a GIST is recognized by a doctor, it's very important that an experienced pathologist, a doctor who studies diseased tissue, becomes involved in making the diagnosis and in studying the characteristics of the disease. So we always emphasize, if at all possible, that the material be examined by an experienced pathologist who has seen and dealt with GIST in the past. So every GIST begins as a localized growth, perhaps in the stomach. If the disease is localized, in favorable cases, surgery alone can be curative. A surgeon will simply remove the localized tumor and the disease is gone. However, that primary GIST, as I've said before, can metastasize, that is, spread to distant sites in the body. That can happen even after an initial surgery that appeared to be entirely successful. The surgeon can never be sure that every single cell is removed. After a GIST has spread in the body, moved to distant sites like the liver or the lungs, then surgery can no longer suffice. Surgery can no longer cure the disease because it's spread throughout the body. That means that systemic therapy, and that usually means drug therapy, is necessary. One of the reasons that an experienced pathologist is important is because once a sample of the primary tumor has been obtained at surgery, it will be examined by the pathologist, and the pathologist can then make an assessment of risk. So if the disease is localized, the pathologist can make an estimate as to how likely it is to metastasize. And as we'll see in a minute, that can affect how the disease is going to be treated. There are two major factors that the pathologist will look at. First of all is just size. How big was the local tumor when it was removed? Bigger is more dangerous, smaller is better. Secondly, the pathologist will look under the microscope at the cells of the tumor. And the pathologist can look at the slide and measure how many mitoses or cell divisions are visible in the tumor. The more mitoses, the more cell divisions, then the faster the tumor is growing. So a higher number is worse prognosis, a smaller number is better. There are detailed quantitative analyses that can be made based on those numbers, but I'm not going to go into those details. Now this matters because it provides an estimate of the danger of metastasis, and that can also affect the course of treatment. As we will talk about a little bit during this seminar, even localized tumor can be treated with what we call adjuvant drug. So adjuvant means that after localized disease, drug treatment is initiated with the goal not of curing the disease, but in fact of preventing recurrence. Delaying or preventing recurrence, that is, another tumor arising at the initial site, or metastasis spread throughout the body. We know now, based on very solid evidence, that this can work. It's not clear how long a course of treatment is appropriate. After all, with adjuvant therapy, the doctor is giving the drug to a patient who does not have any evidence of disease. It's a preventive treatment. So doctors are understandably reluctant to prescribe a drug which has its own side effects and dangers. for a long course of treatment when there was no evidence of disease. But the consensus now is typically 2 or 3 years of treatment. This is an evolving consensus as the evidence accumulates. The higher the risk of metastasis, as assessed by the criteria I just described, size and mitotic rate, the higher the risk, then the greater the motivation is to prescribe adjuvant therapy. For a low-risk tumor, maybe adjuvant therapy is not appropriate. I think we've had that slide already, did it? So, now another important point that is often misunderstood and that I really want people to take home as an important message. When a GIST tumor arises, for example, in the stomach, and then later metastasizes or spreads, that means cells from the original tumor have seeded themselves in other organs, such as the liver, for example, and have begun to grow as metastases in those other organs. But a GIST cell that has moved to the liver and begun to grow in the liver is still a GIST. It's still a GIST because the cells came from the original GIST. It has to be treated as a GIST. This is a common confusion, understandably. So you will hear people with any kind of cancer. For example, a breast cancer patient may say, I've heard breast cancer patients say this, I was diagnosed with breast cancer, but now I've got liver cancer. But in fact, what has happened is they don't have a liver cancer. They have a breast cancer metastasis that has spread to the liver. Similarly, a GIST that has spread to the liver is still a GIST, because the cells that gave rise to that tumor are GIST cells, ICCs. They're not liver cells. And that's a huge difference, because a liver cancer, which would be a cancer arising in the liver, and that happens, a liver cancer is treated completely differently from a GIST. So I hope that that is a crystal clear distinction. So what is the initial treatment then if the GIST has metastasized? Before 2000, we had really no good effective treatments available. Now we do. In about the year 2000, the new drug imatinib, or the brand name is Gleevec, was tested for the first time in GIST and turned out to be a tremendous advance. So the standard first therapy then is treatment with this drug, Gleevec or imatinib, which is beneficial in most, but not all, cases of GIST. Beneficial means, in the best scenario, that the tumor greatly shrinks, begins to disappear. If there are metastases, often what happens is the metastases either shrink or at least they stop growing. So in most cases, there's a favorable response to Gleevec, but not always. And unfortunately, even when the initial response is very good, over time, as happens with many drug therapies in cancer, resistance to the drug can begin to develop, but that's a complicated issue with regard to GIST. We're still trying to learn exactly the implications of resistance. Some or all, usually just some of the metastases may start to regrow. So at that point, we have what is called imatinib-resistant, drug-resistant GIST. So what can be done at that point? Well, there's still a lot of options. Surgery can be helpful. Some of the metastases may be dealt with surgically or by techniques like radiofrequency ablation, which can be talked about later. I won't be talking about that, but you'll hear about that in other presentations probably. Or we can escalate the dose of the drug to see if that helps. Or we can move to what are called second and third line drugs. And we now have a second line drug, Sutent or sunitinib, and a third line drug, Stivarga or regorafenib. Beyond that, there are a lot more drugs in clinical trials. I'll talk about that a bit later. So there are a lot of of options for imatinib-resistant GIST. But once we're dealing with imatinib-resistant GIST, again, I would urge that people are seen by a specialist who really knows and has familiarity with the disease, because it does get complicated. Okay, on to talk about the biology of GIST a little bit more. It was a single landmark discovery made in 1998, and I'm not going to describe how we got to that point, but in 1998 a tremendous discovery was made which completely transformed our understanding of GIST. It was made by a group in Japan, building on the work of scientists in other countries, including Canada, I might say. And the discovery was that most GIST cells express a particular protein, that is, they make a particular protein called KIT. It's a funny name, but it actually comes from kitten, cat, because in fact this protein was originally discovered in a virus that infects cats, but that's another long story we don't need to talk about here. So GIST cells express an unusual protein called KIT. Very few other cells in the body do that. Different kinds of cells in the body express or make different kinds of protein. That's fundamentally why they're different, why a heart red blood cell is different from a heart muscle cell, is different from a skin cell. Different cells make different proteins, and GIST cells usually make this protein called KIT. And why do they make it? They make it because typically what's happened is that there has been a mutation in the DNA of the cell affecting the KIT gene. And so they make, not only do the GIST cells make KIT, but they make an unusual or variant or mutated form of the KIT protein. And this particular form is issuing a command to the cell, telling it to keep dividing. So we say then that the mutation in KIT drives the GIST. It's issuing the constant command to the GIST cell to keep dividing and growing, even though it shouldn't. Now, this is found in most cases of GIST. There are still some GISTs where the KIT protein is expressed, but it is not mutated. It's just the normal form of KIT. A lot of research has gone on to try and figure out what's driving those GISTs if it's not a KIT mutation. In 2003, Jonathan Fletcher and other colleagues discovered that in about 10% of GISTs, where the KIT gene is normal, there is a mutation, but it's not in KIT. It's in a very closely related gene, although it has a very different name. It's a related gene with a very different name. It's called, and this is a mouthful, it's called platelet-derived growth factor receptor alpha, or PDGFRA. So we've now accounted for about 85% of GISTs. It's either a KIT mutation or a PDGFRA mutation, but that still leaves a small percentage, maybe 15%, that are normal for KIT and for PDGFRA. So what's going on there? Well, these GISTs are typically called wild-type GISTs. Wild-type is just the term that has always been used in genetics to refer to the normal gene, a gene that is not mutated. Geneticists always call that the wild-type. So wild-type GIST just means that in these GISTs, there is no detectable mutation in KIT or PDGFRA. Probably wild type is not the right terminology. Probably these GISTs also contain mutations. We just don't know yet what the mutation is. It's probably more accurate than calling them wild type, but still people call them wild type. Wild type GISTs are a rarity within a rarity. They're a small minority of GISTs. Even for these so-called wild type GISTs, a lot of progress is being made. And there was a big discovery in 2011 it was discovered that a lot of these wild-type GISTs have mutations in another gene, completely unexpected mutation, in a gene called SDH, which codes for an enzyme called succinate dehydrogenase. So we now know that a lot of the so-called wild-type GISTs are really SDH mutant GISTs. These are typically GISTs that occur in children or young adults, so-called pediatric GIST. So most GISTs occur in older people. Like most cancers, the incidence goes very much up with age. GIST patients tend to be older, but it's been recognized from the beginning that there are some GISTs occurring in younger people, and these are called pediatric or wild-type GISTs. So as you can tell, it's complicated. In fact, every year we learn more and more complications. One could say now that GIST is not really a single disease, but we already understand that it's really a group of related diseases. They have similar and yet distinct molecular characteristics and clinical response. Well, that's a bad thing because it makes life and medicine more complicated. It makes GIST oncology more complicated, but it's also a good thing because it means that as we learn more and more about the specific, almost personalized characteristics of diseases like GIST, we can hope to focus therapies more carefully, targeting them to exactly a particular kind of GIST. So for the remainder of the seminar, I'm going to talk mainly about KIT mutant GIST because that is far and away the most common form. So I've already been talking about genes and proteins. Let's just say a little bit to explain what we're talking about here. So genes, as I think most people know now, it's pretty common understanding now. Genes are made out of DNA. DNA is the molecule that is the blueprint or the coding information that tells the cell how to make proteins. It does other things too, but it tells cells how to make proteins. Again, as most people are now aware, the complete human genome has been sequenced around 15 years ago now. We know that it encodes at least 30,000, probably more, different proteins. KIT is just one of those. PDGFRA is just one of those many kinds of proteins. So DNA is the code. The code is read out by being converted to another molecule, RNA, that we don't need to discuss here. And the RNA is used to synthesize the protein. The sequence of the protein is dictated by the sequence of the DNA. So a change in the sequence of the DNA, a mutation, leads to a change in the sequence or structure of the protein that it makes. So back to KIT. KIT. It's also sometimes called c-KIT, and it's sometimes called CD117, but these are just different terminology for the same thing. KIT is one particular protein out of the tens of thousands on the human genome. As I said before, there are very few kinds of adult human cells that make KIT. Most cells have the information, but they don't make the protein. And those cells include the normal ICCs in the gut, and they include GIST. The most important step, the most important test to be made in diagnosing GIST, when a sample of the tumor is obtained, the most important thing is that the pathologist tests whether the tumor cells make KIT. Do they, as we say, express KIT protein? How do you know? Well, the pathologist figures that out by doing a specific kind of test. Again, it's got a complicated name. It's called an immunohistochemistry, or IHC test. Essentially what he's doing, or he or she is doing, is to treat the sample of the cells with an antibody, another kind of protein, that recognizes the KIT protein. It's a kind of staining technique. I won't go into further detail about it here. And by looking under the microscope and looking at that staining, the pathologist can determine that the tumor cells express KIT, and that's extremely strong evidence that it's a GIST. Not perfect evidence, because there are a few GISTs that apparently don't make KIT, but it's very strong evidence and very important evidence. So KIT is one of many genes that we call onco or cancer genes. So by an oncogene, we mean it's a gene which, when it's mutated, can give the cell instructions to keep on dividing. The analogy that's sometimes used, and it's a pretty good one, is that the cell is controlled, the growth of the cell is controlled like a car is controlled by stepping on the gas. And you can step on the gas or you can step off the gas. But an oncogene is like a gas pedal that's got stuck. So it's on all the time. Okay, so this just reiterates, I think I'll skip over this slide, just reiterates that in GIST, the KIT gene and is mutated, the protein is abnormal. And this emphasizes again what I think I've already said, that a mutation is a change in the DNA sequence encoding the protein. This is dealt with in other parts of the LifeRaft Group website. I'm going to move on in the interest of time. Also important to emphasize that the mutations which have occurred, which give rise to the GIST, these are mutations that have occurred during a person's life, but they don't affect the germ cells, the egg or the sperm. That is, even though there's a mutation involved, it's not a mutation that can be passed on to your children because it's only occurred in those specific cells in the tumor. Just now, when we say mutations, those mutations can occur at many different points along the KIT gene. And in fact, we've learned now that where that mutation occurs in the KIT gene, along the KIT gene, has a big influence on the biology of the resulting tumor. It's correlated with where the GIST tumor arises. It's correlated with prognosis and response to drugs. So we believe that mutational testing, which can be done by DNA sequencing, should be done on all new GIST cases. However, we're very far from being at a point where that happens, even in the US or Canada, let alone in other countries. So we're urging that more mutational testing be done. And now let's turn to talk about drugs for a few minutes, the development of targeted drugs. So the drugs like Gleevec and Sutent that most of you have heard about or are taking, these drugs are what we call small molecule drugs. That's as distinct from, say, an antibody or a serum or a vaccine. So these are small molecules that are synthesized by organic chemists in a laboratory. They're typically orally bioavailable, that is, you can take them as a pill. They don't need to be injected. Typically, they're what we call enzyme inhibitors. They're compounds that can specifically shut down or turn off a particular enzyme. Many drugs are like this. For example, if you've taken Celebrex for arthritis, that's an inhibitor of an enzyme called cyclooxygenase. So this is a common theme in drug development, developing a small molecule compound that inhibits a particular enzyme. An enzyme is a kind of protein. KIT is a kind of enzyme. It's inhibited by Gleevec. So how did this happen? Well, it turns out, and I'll just mention this briefly, many of you will be aware of it, that there's another apparently unrelated cancer called CML, or chronic myelogenous leukemia. Just as KIT Just as GIST is usually due to a mutation in KIT, CML is usually due to a mutation in a different gene called ABL. And it was discovered in a very important breakthrough around 1996 or so that drugs that inhibit ABL can treat chronic myeloid leukemia. It was a huge breakthrough. It came before the development of drugs for GIST. In fact, if there's any justice in the world, I don't know if there's justice in the world, but if there is any justice in the world, these individuals who received the Lasker Award are soon going to receive the Nobel Prize in Medicine because it was a huge discovery. Brian Drucker and his colleagues who discovered that you could treat CML with Gleevec. What's that? And this led to a huge revolution in cancer treatment, and some of you have seen this book, Magic Cancer Bullet. It made the cover of Time magazine. The development of this drug that inhibits ABL and controls chronic myelogenous leukemia. So what's this got to do with GIST? Well, it turns out that GIST therapy had a lot in common with CML therapy. Why? Because KIT, the gene that often drives GIST, is closely related to ABL, or BCR-ABL, the gene and mutation that drives CML. So many of the drugs that were effective in CML turned out to be effective in GIST. Not all, but many. In fact, and you can't see the details of this, but this is a family tree of a whole lot of related human proteins. It's just like a genealogy tree. And you can see that KIT and PDGFRA are very close cousins. They're almost brother and sister. ABL is on the same branch, same general part of the tree, but it's a more distant cousin. These are a few slides about the mechanism of action of Gleevec in inhibiting GIST, in inhibiting KIT, rather. I'm going to skip over this in the interest of time. It's more detailed than we need. So Gleevec was a breakthrough for CML. In 2000, we learned that it's a breakthrough drug for GIST. In fact, it began a whole wave of new targeted drugs, not only for GIST, not only for CML, but also for many other cancers, including, for example, renal cell cancer in the kidney, NSCLC, which is non-small cell lung cancer, a form of lung cancer. So imatinib was really a new paradigm or a new kind of way of treating cancers. All of these drugs inhibit a particular large class of proteins or enzymes, which we call TKIs. That is, the enzymes are tyrosine kinases. That's the class of enzyme. And the drugs are TKIs, tyrosine kinase inhibitors. So you'll hear that term, TKI. We need more drugs for GIST, and more drugs are being developed. Why? Well, a number of reasons. First, some GISTs are imatinib resistant right from the outset. For example, the most common kind of GIST caused by a PDGFRA mutation, it's a particular mutation. Unfortunately, that particular mutation is resistant to imatinib and also to sunitinib because of the molecular nature of the mutation. Secondly, some individuals just do very badly on imatinib. They get severe side effects. They can't take enough drug to be effective. And thirdly, as we've mentioned before, GISTs tend to become imatinib resistant over time, and then we need new drugs. I've already mentioned this, but I'll just go over it again. There are 3 drugs that are now approved in the USA and in Europe, other countries, that are now approved for GIST therapy. The first line or first choice drug is imatinib, developed by Novartis Pharmaceutical Company in 2001. The second line drug developed by Pfizer, the biggest drug company in the world, is sunitinib or Sutent. And very recently, another big drug company, Bayer, has developed a third line drug called Stivarga or regorafenib. There are lots more on the way. There are a lot of clinical trials. There are so many clinical trials for so many different drugs now that I, for one, can't. I think Jerry Call may be the only person who's actually able to keep track of all these different drugs. And that's good news, that there's a lot more drugs being developed. I'm not going to go over all the details. It goes beyond the scope of this talk. And I think we're getting towards the end of what I wanted to say. So I'll thank you for your attention, and now I guess we can turn to answer some questions. I'm not quite sure how I do that.
Thank you, David. This was a great presentation. We actually do have a number of questions. I'm not quite sure if we're going to be able to address them all, but we will do our best and then see if we can get in touch with individuals after the webcast. And people are welcome to email us afterwards if they have any questions. So the first question is, when a primary tumor is initially found, then how do you know it's the primary and not a secondary tumor?
I mean, that's more of a medical question than I'm really appropriate to answer, but I think the answer is simply that really you just look at it. I mean, from when the surgeon looks at where it arises, I think it's pretty clear that a gist around the stomach is a gist. I think that's pretty evident. I think when you just look at where they occur and what they look at, it's pretty clear whether it's a primary or a metastasis. Although it's certainly true that there are some cancers that metastasize where the primary is never even discovered, the so-called unknown primary cancers. So that does happen. But I think it's usually pretty clear to the surgeon and the pathologist.
Okay, thank you very much. The next question, can dose increase cause anemia?
Well, again, I don't want to get too far into the medical treatment issues, but I think the answer has to be yes. I mean, it's generally true that all side effects of drugs tend to get worse as the dose goes up. So I think it's easily imaginable that you'd have a case where a certain dose of Gleevec, for example, maybe 200 milligrams a day is tolerable, but you get up to 400 or 600 milligrams a day and you start to develop anemia. Of course, there are things that can be done about anemia, like treatment with EPO, which stimulates red blood cell production. But I think, yes, increase in dose typically will lead to an increase in side effects.
Okay, next question. Is the KIT protein a mutation of a normal protein or a protein that normally does not exist?
Okay, I guess what you're asking is, where is the KIT protein normally found? So the KIT protein is found, the gene is found in every cell. Every cell contains the same genome. So the KIT gene is found in every cell. Normally, only a very few cell types make KIT, and they usually make the normal form of KIT. So that includes the ICCs and a few other cells like mast cells in the blood. So it's a protein that normally exists, but only on a very few specialized cell types. So if you look at a skin cell or a liver cell, you'll never find it expressing KIT. They just don't. So it's this protein that normally exists on a few cell types, including ICCs. That's why GISTs almost always express KIT. But in most GISTs, that KIT has been mutated, so it's an abnormal form of KIT that is produced. Not all GISTs show the abnormal form, but 85% or so do.
Okay, the next question is, does the mitotic count matter as much with wild-type GIST?
I can't give an authoritative answer to that. I think the answer is yes, but I don't know how good the evidence is for the relationship between mitotic count and prognosis for wild-type in particular. My guess is it matters a lot, but I can't say for sure.
Okay, I have also a subsequent question from the same person. How do we know how active KIT or the PDGFRA receptor is? which can in turn limit the time GIST cells are in, I think I see G0.
Oh, G0. Okay. Now we're getting into some technical cell biology questions. So I think the answer is that normally in the diagnosis, nobody would try to determine how active the protein was as a protein. The diagnosis that's going to be done is to measure the expression level by the immunohistochemistry technique, and with any luck, also to detect the mutation. And once you know what the mutation is, then that tells you a lot about the activity of the protein. But I don't think anybody ever looks directly, clinically, at the activity of the enzyme directly.
Okay, thank you. Does a higher mitotic rate cause a faster recurrence, or does it just cause the tumor to grow faster?
I think both. Again, I'm cautious about answering the medical diagnostic questions. I'm not the authority on that, but I'm pretty sure that the correlation is both. That a faster mitotic rate means the tumor is growing faster. That means the local tumor will grow faster, and for sure it correlates to an increased or faster recurrence or likelihood of recurrence. I think that's all true.
Thank you. Do you have to do a biopsy to test for KIT, or is there a blood test to look for KIT too?
That's an interesting question. I think generally the answer is that it's either a biopsy or more commonly it's surgery. I think most GISTs aren't biopsied, that you just go in immediately and do the surgery. As to a blood test, I know there are efforts underway now to develop what are so-called liquid biopsies, where you can look in the blood, and what you're really looking at are the very few cells that are being shed from the tumor, but with modern technologies, one can actually pick out those rare cells and test. I've forgotten the original question now. The original question was about the mutational type?
Do you have to do a biopsy to test for KIT, or is there a blood test to look for KIT too?
Yeah. So, yeah, could you do a blood test to look for the KIT? I mean, in principle, you could. I don't think that's done. I'm not quite sure why you would want to, really, because, I mean, once the tumor is diagnosed, I don't think that would usually be done. In principle, I guess it's possible.
All right, next question. If someone has had an adverse reaction to Gleevec, would they ever try it again after Sutent or Stivarga no longer is effective?
Okay, I mean, again, that's a medical question you'd have to discuss with the doctor, because it would depend on the nature of the adverse reaction, I guess. But it does prompt me to say something else, that although imatinib and all of these drugs have side effects for sure, usually they're manageable. I mean, it's easy for me to say because I don't take the drug. But they're manageable in the sense that if the side effects like anemia, if they do develop, typically the doctor can manage them by either prescribing other drugs to control the side effects or by just backing off on the dose of imatinib. That's how they're usually clinically managed. So I guess the question was about going back to imatinib after imatinib. I think the question related to, could you go back to imatinib after resistance to imatinib has already developed? It's a very important question. I think in general the answer is yes, you could. Typically, I think doctors don't. But in principle, you could. And I know some doctors are now thinking about that. One doctor, I remember at one of the New Horizons meetings, GIST meetings, one doctor said that perhaps the best, he said it almost as a riddle, perhaps the best treatment for imatinib-resistant GIST is imatinib. What he meant by that is maybe as you move to another drug like sunitinib, maybe you shouldn't give up on imatinib. Maybe, for example, you could continue to treat with both drugs. And I know there are clinical trials underway now looking at that kind of approach. But now we're getting to the certainly to the limits of my knowledge, probably to the limits of what's known.
David, thank you very much for answering these questions and your time today. We actually have a flood of questions that people have sent in, and I deeply apologize. We cannot get to it today, but we will do our best. The LifeRaft group will do our best to answer your questions privately via email. And also, like I said, if anybody has any other questions, they can contact us after the webcast, and we will do our best to get you in touch with the right person to answer them. So with that, I want to thank everybody for their time today. I want to let you know that this webcast is recorded, and we will be making it available on our website and announcing when that will be available shortly. And I also would like to mention that this is one of many in our webcast series. We do plan on covering other topics. If anybody has interest in other topics, feel free to email the LifeRaft group at liferaft@liferaftgroup.org. And once again, David, thank you very much for your time today. As always, your presentation was very informative, and we thank you for that.
Thank you.
Thank you, everyone.